Hi, my name is Yeon Choi. I'm a postdoc in Seoul National University. Today, I will talk about the chronology of mRNP remodeling. mRNA transports genetic information from DNA to protein, so it is a key element of gene expression. To regulate the mRNA and gene expression, many RNA binding proteins bind to mRNA to formulate messenger ribonucleoprotein, in short, mRNP. From birth to death of an mRNP, it changes protein components because each step is regulated by different type of proteins. Therefore, it is important to know protein composition in each stage of mRNP to understand gene expression. To identify the protein components of mRNP, RNA interactome capture or RIC method is widely used. RIC includes protein-RNA cross-linking, RNP enrichment, and mass spectrometry, resulting in identification of hundreds of RBPs. However, it can only identify proteins from the mixture of all stages. To overcome this limitation, we tried time-resolved capture of mRNP. We can learn which RBPs participate in which steps by adding a temporal dimension to the mRNA interactome data. For example, if we collect mRNPs from the early stage of their life cycle, we can find transcription-related RBPs. On the other hand, late-stage mRNPs would contain translation and decay factors. For the time-resolved capture of mRNP, we first pulse-labeled mRNPs by 4-thiouridine, in short, 4SU. After 10 minutes 4SU labeling, 4SU was replaced by normal uridine and chased for minutes and hours. By controlling the chasing time, we could adjust the age of labeled mRNPs. Then, 4SU labeled mRNPs were specifically captured by UVA cross-linking. Under UVA irradiation, only the 4SU containing RNAs are cross-linked to proteins, and those cross-linked proteins are quantified by mass spec analysis. With the mass spec data, we first checked whether it showed expected mRNA binding dynamics on well-known RBPs. Here, we can see that mRNA transcription-related RBPs are released at the earliest time points, and mRNA processing factors are released slightly later than transcription factors. Next, we could observe mRNA export-related proteins at the intermediate time points. Finally, translation and decay-related factors were released at the late time points. We analyzed mRNA binding dynamics of 736 proteins and classified them into 7 clusters by K-means clustering. Then, we visualized geoterms released at specific time points. Similar to the previous slide, transcription was the first released stage, then mRNA splicing followed. Cytosolic localization and translation-related geoterms were released at the latest time point. Finally, we designed a scheme to find RBPs with novel functions. Based on the annotated geoterms, we generated the expected mRNA binding pattern for each RBP. Then, we compared the expected patterns and observed patterns. Large difference between the expected pattern and the actual observation implies unknown function of RBP. To predict expected patterns of RBPs, we started from the gene versus geo Boolean table. Then, this table was converted into numerical vectors by the multiple correspondence analysis. Finally, for each time point, a rich regression model was fitted to predict protein quantity Z-scores from geoterms. Previously well-studied RBPs such as RNA polymerase II and eIF4G1 showed a nice match between expected and observed patterns. On the other hand, less studied RBPs, determined by the number of PubMed publications here, showed higher prediction error. CCDC86 is one of the RBPs whose dynamics is not consistent with their reported functions. It has been reported to nuclear localized, but our observation indicates its function in the cytoplasm. So next, we measured RNA level and protein synthesis level after CCDC86 knockdown to find its function. After CCDC86 knockdown, RNA population was nearly unchanged, but protein synthesis was reduced globally. Therefore, CCDC86 may function as a general translational regulator. In summary, we generated time-resolved mRNP proteome data throughout the life cycle of mRNA, and it is largely consistent with previously studied features of RBPs. But interestingly, under-characterized RBPs were systematically screened by geo-based prediction of mRNA binding dynamics. And from this, we suggest a novel function of CCDC86 as a translational regulator. If you are interested, find out longer version of this talk at iRNA Cousin. Thank you for listening.